Today we are going to discuss about vectors, cloning vectors. So a cloning vector is a one which can carry, it's a DNA material which can carry to its host cell, which can carry a foreign DNA material to its host cell. How the cloning vector should be, identical cloning vector should be. It should be small in size. It should self-replicate itself. It should have self restriction site for insertion of DNA, and it should have a selectable markers for antibiotic resistance gene, and also it should be a small in size so that it can easily integrate into a host cell, and also it should have multiple cloning sites, and it should work in prokaryotes as well as eukaryotes. Now we are going to see some of the vectors like plasmids, cosmids, bacteriophages, bacterial artificial chromosome, human artificial chromosome, and yeast artificial chromosome. Now we are going to see first about plasmids. Plasmids are the extra chromosomal ring in a bacterial chromosome. These are the first vectors to use in gene cloning. They are found in bacteria, eukaryotes, and also some other self-replicating DNA molecules. They have antibiotic resistance genes. They have a origin of replication site. The best example for this is PBR 323, that is plasmid Bolivar Rodriguez 322. And now we are going to talk about cosmids. A plasmid with a cos site. Cos site is nothing but a bacteriophage site. A plasmid which included with a cos site, like a round which has been expanded with a spring, like that. We can think like that. A plasmid with a cos site is known as cosmid. So these cosmids are also used to carry a large amount of DNA from one cell to its host cell, and it can self-replicate itself. And again we can see about bacterial artificial chromosome. These are similar to the E. coli plasmid vectors, but here we can see these can be naturally occurred in F1 plasmids. F1 plasmid. They can be used to study genetic disorders, and also this sequence we can accommodate a large DNA sequence without any risk. So the same way with yeast artificial chromosome and human artificial chromosome. Here we can accommodate large amount of DNA molecule into its host cell. So now we can see, for example, like we had seen plasmids, cosmids, phases, bacteriophages also we can accommodate whatever the molecule amount of host DNA we can accommodate in it. So if you have any comments, please comment in comment box. Thank you.